Welcome back. It's Miss Roman, and this is part 2 of the review for the Chapter 10 test. Last video, we ended with our discussion on what light is and the electromagnetic spectrum and the energy associated with photons with different wavelengths. Scientists originally knew that photosynthesis was converting light energy from the sun into chemical energy. And once they learned that the energy coming from the sun came in many different wavelengths, the next question became, well, do plants convert all the photons from the sun into chemical energy, or do plants only absorb certain wavelengths of light? And Englemann designed an experiment to test to see if certain photosynthetic organisms were absorbing one wavelength of photon, multiple wavelengths of photons, all the wavelengths of photons that were coming from the sun. So he didn't have a spectrophotometer or anything like that, but he did know that the products of photosynthesis are oxygen and sugar. And he had bacteria that needed oxygen to live and glucose as a food supply. So what he did was he took some algae and put the algae on a microscope slide, and then he put the bacteria on the slide as well. And when he would shine white light, so the mixture of all of the wavelengths of light in the visible range, when he would shine that light onto the algae would undergo photosynthesis and supply oxygen and food for the bacteria. Then he took his light source and then used a prism to divide the white light up into the different wavelengths of light. And so a rainbow results. And then he used that as a light source for his bacteria and algae on the microscope slide. And over time, he noticed that the bacteria moved. And the bacteria consistently moved to particular regions on the microscope slide that corresponded with different colors. And when he observed this under the microscope, he determined that the algae were only absorbing particular wavelengths of light. And if they did absorb that wavelength of light, then photosynthesis occurred and oxygen and sugar were the products. And the bacteria would move to that region so they could get oxygen and some food. And Englemann, his experiment now looks like the type of data we would collect from a spectrophotometer. A spectrophotometer shines light onto a sample and determines which wavelengths are absorbed and which wavelengths aren't absorbed. And if we were to put a sample in our spectrophotometer with chloroplast from different organisms, depending on what pigments were present, we would see these types of curves, which indicate where light is being absorbed and where light is not being absorbed. On the test, you'll need to know general information about the Englemann experiment. So what was the purpose of the Englemann experiment? The purpose was to determine if photosynthetic organisms were absorbing multiple wavelengths of photons of light or were they only absorbing a few? Were they absorbing all? So basically, which photons are being absorbed during photosynthesis? The experimental design. You'll need to know that Englemann used a prism to take white light and divide it up into the different wavelengths. And you'll need to know that he used algae as the photosynthetic organism. He could have used something else. And then you do need to know that he used bacteria as a way to track where photosynthesis was occurring. And it is the production of oxygen and glucose that the bacteria will migrate towards. So he had to make sure he was using bacteria that needed oxygen, lived on oxygen, and lived on glucose. If he would have used anaerobic bacteria, then this would not have been such a great experiment, because the bacteria, if they don't need oxygen to survive, then if there is or is not oxygen, it's not going to affect the bacteria at all. So he had to make sure that he was using an organism first that underwent photosynthesis, and then he needed the second organism to need the products of photosynthesis. The results were that not all photons are absorbed during photosynthesis. And it depends on the organism as to which photons are absorbed. Most plants have chlorophyll, so they will absorb in this blue and yellow-orange regions. However, accessory pigments will extend the region of photons that are absorbed. So some plants are red or yellow or blue, and that's because they have accessory pigments other than chlorophyll. All right, so chloroplast. You do need to know the structure of the chloroplast. Specifically, it does have a double membrane, and then it has a third membrane, which is called the thylakoid membrane. And the thylakoid membrane is not connected to the inner membrane. Remember, with mitochondria, their inner membrane folded in and made the cristae folds. But in the chloroplast, we do have this highly folded inner membrane substance, but it is a separate membrane. It's not connected to the inner membrane, and it's called the thylakoid membrane. And I put a little checklist or table here for you that is probably a good idea for your cheat sheet for the test, to have still information about mitochondria and cellular respiration, so that you don't get anything confused. Remembering that the stroma is the cytoplasm in the chloroplast, and in mitochondria, it was the matrix. The thylakoid lumen in the chloroplast is the inside of this thylakoid membrane, and it's also called the thylakoid space. So either term, lumen or space, that does mean the inside of the thylakoid. In the mitochondria, we had the inner membrane space, and that was the space between the inner and outer membranes. And both the thylakoid lumen, thylakoid space, and the inner membrane space, they have in common their function. In those spaces, that's where the proton concentrations were increasing to create the concentration gradient, so that the protons can move through ATP synthase by passive diffusion and power an ATP synthase pump. So that is the same, that function that they serve. All right, we also have redox reactions in photosynthesis, and the terms are all the same. So reduction would be gaining electrons, and oxidation would be losing electrons. And a reducing agent would be causing reduction on another molecule, which means it would be giving the other molecule or atom electrons, and itself would be oxidized. So a reducing agent is oxidized as it's reducing another molecule or atom. An oxidizing agent is a molecule that oxidizes a second molecule. And in order to oxidize another molecule, it has to remove electrons from that molecule or atom, which in turn reduces the oxidizing agent. So the oxidizing agent, by just doing its job, it will be reduced, and the second molecule will be oxidized. And the redox reactions that we learned about in cellular respiration, remember, cellular respiration is a process that breaks molecules open and removes energy. And that energy is going to be in the form of high energy electrons. So it is a process of removing electrons from molecules in general. So that would mean we are oxidizing molecules in that process. So the goal or the purpose of cellular respiration is to oxidize glucose, and in the process regenerate ATP. So in the mitochondria and in cellular respiration, oxidation is the target, the theme, even though there's still both redox, but there's still reduction and oxidation reactions occurring, but the overall goal is the oxidation of glucose. And in photosynthesis, it's the opposite. We are putting energy into electrons and then storing those electrons inside of molecules like glucose, other carbohydrates, etc., cellulose. So we are putting high energy electrons into molecules. So those molecules are being reduced. So photosynthesis is a focus on reduction. We are going to reduce carbon dioxide into glucose. Cellular respiration was the opposite. We were oxidizing glucose into carbon dioxide. So once again, redox reactions are happening in both. We have reduction and oxidation. It's just the overall goal of photosynthesis is reduction. The overall goal of cellular respiration is oxidation. Hopefully I said that correct. I'm going to say it again. In photosynthesis, the overall goal is reduction. In cellular respiration, the overall goal is oxidation. All right. Photosynthesis has two stages. First are the light reactions, and in general, that's where photons are trapped, and the energy from the photon is converted into chemical energy. And then, in general, the second part, the Calvin cycle, is storing those high energy electrons inside of a molecule. So building a molecule, reducing atoms of molecules so that we have a high energy molecule with energy stored in it. And first thing we're going to do is watch a short video of the whole process. And this is from the textbook, or the publishers of the textbook. They also have a lot of really, really good videos. So we'll watch this, all of it, and then we'll look at the specific reactions, and then we'll look at the whole process again.
In plants, photosynthesis occurs in specialized organelles called chloroplasts. The internal membranes of chloroplasts are organized into sacs called thylakoids. Surrounding the thylakoid membrane system is a semi-liquid substance called stroma. Photosynthetic pigments are clustered together to form photosystems. When a photon of light strikes the reaction center of photosystem 2, it excites an electron. Two water molecules bind to an enzyme at the reaction center. This enzyme splits the water and uses the electrons from the water to replace the electrons removed from the reaction center. Oxygen is produced in this process. The primary electron acceptor for the light-energized electrons leaving photosystem 2 is plastoquinone. The reduced plastoquinone passes the excited electrons to a proton pump embedded in the membrane called the B6F complex. Arrival of the energetic electrons causes the B6F complex to pump protons from the stroma into the thylakoid space, thereby generating a proton gradient across the membrane. Because the thylakoid membrane is impermeable to protons, the protons in the stroma must pass through the channels provided by ATP synthase. As protons pass through, ADP is phosphorylated to ATP and released into the stroma. This process for making ATP is referred to as photophosphorylation. When photosystem 1 absorbs a photon of light, its reaction center passes high-energy electrons to ferredoxin. The enzyme NADP reductase then transfers the electrons to NADP to form NADPH. Electrons lost from photosystem 1 are replaced by electrons generated from photosystem 2. A small protein called plastocyanin, PC, then carries the electrons from the B6F complex to photosystem 1.
All right, this image is from the Khan Academy, and they have a lot of really good images as well. And this shows the pathway of the electron transport chain, and then also ATP synthase again. So some of this looks familiar because it is very similar to what we saw in cellular respiration. So in the mitochondria, in the mitochondria and in the cristae, we had these embedded proteins that acted as an electron transport chain to actively pump protons into a region that would then, by passive diffusion, move through the ATP synthase channel protein and enzyme, and then that would phosphorylate ADP and produce a lot of ATP. So in the chloroplast, in the thylakoid membrane, we have a similar process. And this process is also creating ATP and some high energy electrons to be used in the second part of photosynthesis. Remember that chloroplasts are believed to have been their own unicellular organism at one time. So they needed to provide their own energy for different reactions. So the fact that the chloroplast has its own built-in power supply to build the glucose molecules, there can be several reasons that it has this, but it does give supporting data to that theory that this organelle used to be its own organism, because it is making its own ATP to build this glucose and cellulose, etc., these carbohydrates. So where you're going to make a mistake is confusing this with oxidative phosphorylation in cellular respiration. So be careful, read questions very carefully. And in general, what is happening in the light reactions is the trapping of photons. There is also a process that is setting up a concentration gradient of protons. And then the electrons are also being added to our electron carriers. And then the electron carriers and the ATP that's synthesized will move to the Calvin cycle, which is in the stroma. Questions on the test, you'll need to know those general ideas. We have active transport, we have passive transport, we have an electron transport chain, we have the electron carriers present, and then you also need to know some very specific information. And specific information you need to know. You need to know that photosystem 2 comes before photosystem 1 in this process. Photosystem 2 captures photons first, and as a result, it will eject electrons that will travel to photosystem 1. So the electrons that arrive in photosystem 1 are coming from photosystem 2. And in between, they travel through an electron transport chain that is actively pumping protons into the thylakoid lumen. A way to tell the difference between photosystem 2 and photosystem 1 in the laboratory is that the chlorophyll molecule in photosystem 2 absorbs greatest at 680 nanometers, so it's called P680. And the chlorophyll molecule in photosystem 1, that's in the reaction center, is a chlorophyll A molecule, and it absorbs strongest at 700 nanometers. So the chlorophyll A molecule in photosystem 1 is called P700. And know that the concentration of protons increases in the thylakoid lumen. And know that in the thylakoid lumen, we also have water breaking. It's called hydrolysis. Water breaking, and what that means is the water molecule is donating 2 electrons to the chlorophyll molecule in the reaction center, so P680. Water will replace the electrons that P680 releases into the electron transport chain. And when that happens, the byproduct is oxygen gas and some more protons. So when water splits, when water is donating electrons to P680, it is also producing protons. So we really have 2 supplies of protons in the thylakoid lumen. And it's important to know that oxygen is generated at the very beginning of photosynthesis. Remember that we didn't see oxygen till the end of cellular respiration, but oxygen is actually produced at the very, very beginning of photosynthesis. By the end of photosynthesis, we have electrons that are being carried by our electron carrier to the Calvin cycle. And these high energy electrons have high energy because they absorbed energy from the photons. And I have another video for us to watch, and then we're going to talk a little bit more about how the photons are absorbed, how that energy gets into electrons, and then how those electrons are able to then travel and take the energy with them.
The leaves of this plant have cells that carry out photosynthesis. If we zoom in on this photosynthetic plant cell, we can see the chloroplast, where the reactions of photosynthesis occur. Photosynthesis consists of two primary steps, the light reactions and the Calvin cycle reactions. In this tutorial, we'll focus exclusively on the light reactions. The light reactions occur within the thylakoid of the chloroplast. Here, special pigments absorb light energy and transfer it to high-energy electrons, eventually producing ATP and the electron carrier NADPH. Let's zoom into the thylakoids to take a closer look at how ATP is created. The light reactions use two photosystems, called photosystem 1 and photosystem 2, which are both embedded in the thylakoid membrane. It's important to realize that these photosystems are named for the order in which they were discovered, not for the order in which they participate in the photosynthetic process. The light reactions actually begin at photosystem 2. The first thing that happens is that photosystem 2 receives photons, or light energy. This light energy is transferred to a chlorophyll reaction center, causing electrons in the reaction center to become energized. These electrons become so energized that they escape photosystem 2 and move to a nearby electron acceptor molecule located in the electron transport chain. Meanwhile, to replace the electrons leaving photosystem 2, water is split, releasing oxygen, 2 hydrogen ions, and 2 electrons. The first set of electrons continues to move down the electron transport chain, releasing stored energy as it moves. This energy is used to create a hydrogen ion gradient. A protein in the electron transport chain pumps hydrogen ions from the stroma into the thylakoid space. This creates a high concentration of ions in the thylakoid space relative to the low concentration of ions in the stroma. This gradient contains a large amount of potential energy, which is used by an enzyme called ATP synthase. The hydrogen ions flow down their concentration gradient through a channel in ATP synthase, releasing energy in the process. ATP synthase uses this energy to add a phosphate to ADP, forming ATP. Let's zoom back out for a moment and return to our chloroplast. Remember from the beginning of this tutorial that the light reactions produce both ATP and NADPH. We've just seen how ATP is produced, but what about NADPH? Let's zoom back in to take a closer look. Notice these electrons over here where we left them at photosystem 1. As photosystem 1 absorbs additional light energy, the electrons again become energized, escaping photosystem 1 and moving down the second electron transport chain. Electrons from the electron transport chain adjacent to photosystem 2 replace those in photosystem 1. And again, water is split to replace the electrons that have moved from photosystem 2. At the end of this electron transport chain, the energized electrons and a hydrogen molecule are used to reduce NADP to NADPH. Let's zoom back out to review. The light reactions use light energy and water to produce ATP and NADPH. Oxygen gas is released as a byproduct. Together, the ATP and NADPH formed during the light reactions are used by the Calvin cycle reactions, which are discussed more in depth in a separate tutorial. The important thing to remember from this tutorial is that plants need both light and water to survive. Without these ingredients, the light reactions would shut down, stalling photosynthesis and causing the plant to die.
All right, that is a really good description of the light reactions in photosynthesis. Ricochet Science, they also make a lot of really, really good videos that really focus on what you need to know and what's important about these systems. So let's look at this picture of the photosystem a little bit more. We need to spend a little extra time on the photosystems themselves because you do need to understand this energy transfer that's happening. All right, we'll actually stop here and pick up in part 3. I don't want this video to get too long. So we really don't have that much left to go. So I will see you in video number 3.